Okay, recording. So, yeah, today's workshop. Jordan, do you want to introduce it?
Yeah.
Sure. Oh,
Jordan.
Yeah.
Sorry.
Okay,
No, go ahead. Yeah, you know more than me. I'll just say, thanks everyone, Francisco, for coming. We had a few technical difficulties, but we're really fortunate to have Jonah here teaching us, and I'll let you take it from there.
Yeah, thanks. So I'm going to talk a little bit about sort of analyzing population genetic data using a couple of unsupervised clustering methods. And I'll introduce sort of why we might be interested in studying genetics, studying large-scale population genetic type data, and then go through sort of like the implications that you get from finding patterns in the data, how it's sometimes misinterpreted, and what it can actually tell you. And in explaining this, I'll actually go through some of the algorithms that are used. Oh, this is host disabled participant screen sharing. So you need to change that.
Nick, you're muted.
Sorry, yeah, you're all good, Jonah.
Oh, there it is. Cool. So you can see my PowerPoint screen. Okay. Yeah, so just a brief outline. I'm going to talk about genetic variation, how we measure it, how it's represented typically in a file format, and the type of patterns you can find with looking at it. I'll talk about population stratification and principal component analysis. This will be done through a live demonstration in RStudio. If you don't know what principal component analysis is, that's fine. I'll explain it quickly. Oh, right, and I have a Dropbox link that I can give you that has my slides and the script that I'm going to run through. So let me pop open the chat real quick and paste that. Okay. Okay. So a quick primer on genetics. So between two people, there are about 20 million different genetic differences. While this seems like a lot, it only accounts for about 0.6% of our 2.6 billion base pair genomes. Probably you learned in high school at some point that variants are passed from parent to offspring through DNA. Of course, people have observed that parents or kids tend to look like their parents probably way longer than we knew anything about DNA or about genetics in general. But the reason that we see a sort of hereditary nature of traits is because, of course, variants are passed from parent to offspring. And there's patterns that can exist in these variants that we can find. These patterns can tell you about your genetic ancestry and can predict different disease statuses plus other traits. So this goes into what we would typically think of as genome-wide association studies, which I won't really talk about beyond this, but the goal of a lot of genetics really is trying to predict traits from the genetic differences we see in the population. Just another quick primer, different types of genetic variants are single nucleotide variants. Again, where you just have a single difference between different individuals. So in this diagram, of course, this is like the genomes of 4 different people, and there can be 1 or 2 small differences. There's insertions and deletions, where you can have multiple base pairs of DNA cut in or cut out. Like you can see there's a C that's been inserted, and here there's 2 base pairs or 3 base pairs that can be removed, which can, of course, cause different problems. And there's more complicated structural variants where like large sections of the DNA are cut out or flipped or like completely or repeated multiple times. Those are a little bit harder to find and kind of beyond the scope of today. How are variants detected? Of course. So maybe the one that's most commonly used, and if you've ever gotten any kind of genetic testing done, either through like 23andMe or through like a test for a particular variant that you may or may not carry, is probably done through a SNP microarray. So the way this works, if you provide a spit sample, in the case of 23andMe, they chop up the DNA to little tiny pieces, and then there's basically a chip of silicon with lots of different probes on it. These probes correspond to different variants that you may or may not have. And because you can separate the DNA out and then try to separate the 2 strands, and then if the DNA will bind to one of the probes on the chip, at a particular locus, it'll emit a light impulse. And if it doesn't bind, then you won't get a light impulse from that. And so if it does bind, that corresponds to having the variant, and if it doesn't, then you don't have it. So this is pretty cheap and really accurate. It can provide a lot of useful information, but it only covers a very small amount of the genome. Basically only covers the amount that you actually have probes for. So if you have, so a lot of probe sets have like, or SNP chips have like 100,000 something probes, which again is quite a lot of data, but it's a very small fraction of the whole genome. But if you want a lot more coverage, if you want to sequence everything, you can use high throughput sequencing, which is like basically gives you everything. Well, can tell you all the variants you could possibly have, which is a lot more expensive, but of course we know it's getting cheaper. I'm sure you've seen that graph that shows you how much sequencing costs have decreased over the past, I don't know, 20 so years. And this gives you everything. It gives you the non-coding variants, coding variants, parts of the genome that basically show no variation at all. And while this is really good, like it's what I use for most of my research is whole genome sequencing based, it is kind of overkill for a lot of applications. Because we have so much data, this is where data science can really come into play. So one thing that's important to know about genetic data is that it's extremely high dimensional. High dimensional means that there's lots of features, so many SNPs or single nucleotide polymorphisms, and much fewer samples. So again, going back to like, there's a few 100 million differences in the genome. You usually have a lot fewer than 100 million samples that you're working with. Of course, now there are some biobanks that are getting up into that number of people. But for the most part, we're going to have a lot more features than we do people that we can actually analyze. And the features are often correlated. So what I mean by that is that there's some variants that you will always find in groups. Like, and that's what's referred to as linkage disequilibrium. When kind of like you'll have a couple of variants that are on the same chromosome, or usually a block of a few thousand variants, and they're kind of always inherited as a group. And that correlation structure is really what a lot of machine learning algorithms are trying to find. Like if you have a feature set of 1,000 features or 10,000 features or 100,000 features, and you want to be able to say something meaningful about all that information, it's really, what you're really trying to do is try to condense that feature space down and find features that kind of give you the same information. Just to preface what we're trying to do. And this is what calls for unsupervised analysis. So maybe, I'm sure you guys probably talked in the data science club at some point about the differences between supervised and unsupervised machine learning algorithms. A supervised algorithm is something where you're trying to predict a label. Like, you know, if you have an image of like handwritten digits and you're trying to predict what digit is this. But with genetic data, we don't really have labels that we're trying to find. We're just trying to find some patterns in the data. And that's what's referred to as unsupervised analysis, or unsupervised algorithm. When you don't really know what the labels you're looking for are, you just want to see, is there any kind of structure? So what we're going to use, at least in the demo, is what's called principal component analysis, or PCA. And a PCA is used kind of like all over the place. It's a really kind of workhorse algorithm that it takes high dimensional data and compresses it down to a smaller feature space that shows you the major differences in the data. And in this little graphic, this is just like a little visualization of what PCA is doing. Of course, in this example, there's lots of samples and only 2 features. And the PCA algorithm will try to find these 2 vectors that kind of explain the most variation in the data. This is really a simplified version, well, a simplified kind of example. Of course, with genetic data, we have tons and tons of features, many thousands of dimensions, and much fewer features, or much fewer samples. But this is kind of the idea behind what it's doing. So I'll move on to the demonstration now, but just to introduce what we're going to look at, we're taking some data from the Human Genome Diversity Project. This is a consolidated data set of human genomes from about 51 populations around the world. And kind of the goal of this project was to map out all the major differences that you find just in different human genomes. The data set's really huge. I think in the R script, you can actually download the entire thing. It's about 2 gigabytes. But I provided a small subset of the data just because it's much like your laptop probably won't be able to handle doing a 2 gigabyte file PCA, or it will take a while. Just for the purposes of this demonstration, to give you a small slice of it. So we're going to run PCA to see if we can find some patterns in this data. And then we're also going to look at, well, the first thing we're going to do is look at the correlation of different SNPs within this small subset of data. And the way you measure LD is with this thing called the D prime score. And I started to give you a quick diagram of what D prime actually tells you. But basically, imagine this is a strand of DNA, and these are 2 variants, variant A and variant B. And the probability of having variant A is maybe it's 20%, like 20% of the population carries this variant, and variant B is 30% of the population. And if these variants are independent, like there's no, like this is just some basic like conditional independence stuff from your intro stats course you may have taken at some point. But if you have the probability of A and probability of B, if they're independent, then inheriting them together will just be the product of the 2 probabilities. But if there is some correlation structure here, if you're more likely to have variant A when you also have variant B, then there's going to be a difference, right? And that's what's referred to as D, the number. And so for every pair of variants in the genome, you can calculate this D score. And then the D prime score is used to normalize it because if you can do the mental math and imagine different variants with different allele frequencies, this D score will be different depending on the allele frequencies of the different variants. So by taking the maximum possible D value, that kind of normalizes it, so it's easier to compare different variants to each other, or different pairs of variants and their LD scores.
So how do you find the D max?
So that would be like, well, the maximum D score, I guess, would be the product of the 2 variant allele frequencies.
Okay.
Right? If this is, yeah, like if they're completely uncorrelated.
Would you take all pairwise D prime scores and get the max of that?
All pairwise.
Yeah, I'm assuming that D max would be like the maximum D prime score for any variant.
Yeah, well, Dmax this is like for any pair of variants. It's just a normalization factor, so it's easier to compare.
Okay.
Gotcha.
Yeah, because if you have different variants with different frequencies, the maximum D score will be different.
Oh, yeah, right.
So
the maximum is if they are perfectly correlated, so that the PAV is 1 minus PA times PV. Does that make sense?
PA.
If they're completely uncorrelated, it's 0, right? The D. Because PAV equals PA times PV, so the DAV will be 0, right? So I guess the D max should be when PAV is 1, right? Probability of having it to change is 1.
Yeah, like if they're always perfectly correlated. maybe I'm butchering this explanation. Like I know there's a lot of literature on why you would like want to normalize by Dmax, but I think it's the maximum possible D-score given the allele frequencies.
Okay.
So it would be like the product of, yeah.
Cool.
But again, like a lot of things that it's implemented in a package in R, which makes it easier to not have to actually know how it works. But which I try to avoid doing that because I would like to actually understand how the method is exactly done.
Yeah.
We'll see. Okay, so I'm going to switch over to my R Studio session right now. R Studio share. Okay. You guys can see this?
Yes.
Okay, cool. So if you downloaded the data and followed along with me, it should just run out of the box. Or if you just want to follow along here, can you guys? Is the font size okay too? Should I make it bigger?
It's okay for me.
Okay, cool. Yeah, so data comes from Human Genome Diversity Project. You can check it out here if you want to learn more about it. Of course, in R is very modular. You can add packages that help do with the thing that you want to do. So I'm going to use this package called genetics. And if you don't already have it, you can just do install.packages genetics, paste that into the console to take a few seconds to download and install. The next thing I'm going to do is load in the actual data. And this is in a file called HDDP data JE. JE, that's me. I subsetted a small piece of the whole Human Genome Diversity Project data just for sort of ease of use. And right data path. I don't know, this is a little R hack or an R Studio hack, but it's nice. Like this will tell you the location of this, where the script is saved. So instead of having to go through and like try to figure out where the path is and then manually paste it in, this will just automatically grab the path for you.
That's pretty cool. Yeah.
Nice.
And then to read in the data, you use read.delim. So now you can see this object HGDP, or not type, that's Python, class HGDP. It's a data frame. If you take just take a quick look at what the data actually looks like, this is another cool thing you can do in RStudio. You can use view, which will bring up sort of like a Excel spreadsheet kind of representation of your data. Just taking a quick look at it. Every row is one individual, and every column is a feature. So the features, these are different SNP IDs. And typically SNPs, they're represented by this sort of numeric identifier. It's called an RSID. And then the actual data point is the two alleles that this person carries of this SNP. So whatever individual, the first individual at this particular site has a T and a C, right? Because you have two copies of every gene, one you get from each of your parents. And yeah, so AA means that they would have two copies of the A allele. GG means they have two copies of the G allele at this site, etc.
So does this data, come from a SNP array, or is it from NextGen?
I think this is from a SNP array.
SNP array.
Yeah.
Cool.
Yeah, that's typically what the RSID is for. Also, it sort of identifies the probe that goes for that particular SNP.
Cool.
And so again, it's quite a bit of data, but a lot less coverage than you'd get from whole genome sequencing. But I think they designed this probe set to try to capture the most variation between different populations. Yeah, we can take this a look. Oh, we kind of already did that. So the first thing I'm going to show you how to do is just calculate linkage disequilibrium using another package. right, which tells you how correlated genetic variants are in the population. So it's kind of one quick way to look at like if there's some structure here at all. So this is just some R code hacking. But one thing I wanted to do was, it doesn't really make sense to calculate LD between variants that are on different chromosomes, because like the way that like variants are inherited together on one contiguous strand of DNA, and then through the process of crossing over, you can get variants to kind of be split up in genealogical time. So I just wanted to grab all the variants that I have that are on chromosome 1. Lane. So I got about 70 SNPs we can calculate LD for. So that's a 70 by 70 matrix of LD scores. Here's just some more hacking you can follow along with, but because we have this, one of the things in the genotype packet or in the genetics package is you can convert these AA and AT, these like two letter strings, into a type called genotype, a data type called genotype. That's another cool thing about R is you can kind of define your own data type, because you have strings and factors and numerics and whatever, but you can also define a new kind of data type. And so in this genetics package, they make a genotype data type, which now you can see instead of just the letters, they're separated by a slash. And this kind of genotypes. If we look at this, they're now, it has the actual like genotype itself, but it also has information about the two different alleles. So it's kind of like a factor data type, but with a little bit more information. And when you're working with genetic data, it can be helpful to convert the actual SNP array data into this genotype format.
That's really cool.
Yeah. Okay. And so now there's this convenient function called LD. Again, all the magic happens under the hood, so you don't actually have to know what's going on, which is nice. It's going to take a few seconds because it's a lot of data to turn through.
So what exactly is the LD mat doing?
So it's calculating this D prime score between every pair of genotypes.
Okay.
Do you know if it's parallelized at all? Genetics?
I don't think this is doing anything in parallel. Okay, that took like.
That's what I think.
Yeah,
so.
this is, and maybe I could speed this up by doing parallelization. Now, if you look at LD mat, oops. It's another new data type, LD data frame. So it's like a data frame, but it has a little bit more information. Yeah, so basically it's a square matrix, but it also has these different, so it's like, this is the D score, D prime score correlation. You can also measure LD by a chi-squared value. And those are all the different ways of measuring LD. And then if you use the LD heat map package, which again, if you don't have it installed, you can just do a quick install.packages, LD heat map. And it'll draw this cool looking graph or kind of, heat map structure. Sorry, this is taking a little while.
No problem.
Be patient. Oh, it'll happen.
Who developed
Why
that?
is it taking so long? What's up?
Who developed genetics package?
I'm not really sure.
There it is.
Probably some of those like people at Harvard that do a lot of like Mark Daly's group.
Yeah.
Yeah.
Did a lot of the like kind of like hardcore population genetic stuff. Yeah. So this is what an pairwise LD matrix typically looks like. Now, because I just grabbed variants all spread out across chromosome 1, you don't really get to see the structure the same way that you typically would. What's cool about LD is that it forms these block-like structures. So if you had lots of variants that were close together, you would see the variants that are all really correlated in one particular area. And then you would see another block of LD in another area of the genome. But it forms these sort of like blocks.
Mm-hmm.
So for the purpose of this demonstration, I kind of just randomly chose SNPs. So you're not really seeing the structure that you'd like to see, but there's lots of places you can go to actually like just look up LD segments across different parts of the genome. Okay. But that's kind of cool. But that's not really like the most cool thing we're going to talk about.
Mm-hmm.
So really what kind of the meat of this presentation is to look at how principal component analysis can be used to measure genotype data. So we have this HGDP data frame. And so the first thing we're going to want to do if you to run PCA is you need to get it into a like machine learning ready format, right? Because you can't put AT and AA and AC, whatever, into a machine learning algorithm. Like it needs to have some kind of numeric representation. So R has this really nice data matrix function. So now if you look at HGDP, well, genotype, now instead of letters, it's just represented by numbers.
Mm-hmm.
So because when we converted this data into the genotype data type, it's basically a factor representation. So now 1 would be like the first factor, 2 would be the second factor, 3 would be the third factor, right? Because there's 3 possible genotypes you could have. You could be like homozygous for the reference, heterozygous, or homozygous for the alternative allele. And so that's what 1, 2, and 3 represent here. And so now that you have a numerical representation of the genetic data, now you can just run PCA. And in R, the function that does principal component analysis is called PRComp.
Mm-hmm.
And it's typical to center and scale your data first. That just means you're gonna set it to a mean of 0 and a standard deviation of 1. If you want to learn more about how PCA works, you know why that's an important thing to do. But just believe me right now that you should do that. And again, this will take a few seconds because, okay, that was faster than the other one. So once you run PRComp, well, that's not what I want to do. So the output of PRComp, the principal component analysis function, is basically a list that gives of a couple of different things. It gives you the standard deviation. So if you learn a little bit more about how principal component analysis works, it's doing what's called a singular value decomposition of the data matrix, which means it decomposes, it splits the matrix into 3 smaller matrices. The first one is standard deviation. So this is a matrix or a sort of a diagonal matrix of eigenvalues. If you remember linear algebra, eigenvalue is like particular value that when you do a matrix transformation, it explains the most amount of variation. You have the rotation matrix, which is a matrix of loadings, of PCA loadings, which again, it's like a vector that transforms the data into another feature space that explains the most variation. And you have the X, this X data frame, right?
Mm-hmm.
Which is the actual principal components that tell you information about the data. Okay. So just remember before we go through this, is like these are just 1,000 randomly sampled variants from this data set. And when you run PCA on them and then plot the first 2 columns of this X matrix that you get out of PRComp, you get this really distinctive structure. So, and again, this is just like 1,000 random SNPs that I picked.
Right.
And so what do the axes tell you here? So usually when you want to plot principal components, you'll plot the first principal component and the second one. And the way I sort of think about this is that the first principal component explains the most amount of information in this lower dimensional feature space. And then the second principal component explains the second lowest amount of, or highest amount of information. And then basically you just go down from there. So the third, fourth, and fifth principal component explain less and less of the variation in the data. And the way that you calculate the percentage of the information that's being explained is you, well, you take the square of the eigenvalue or the standard deviation vector that gives you the eigenvalues, and then the magnitude of the eigenvalues is proportional to the amount of information that they explain. So you can sum them and divide by, basically you can normalize the eigenvalues to give you the percent variation explained. And so a lot of times when you're looking at principal component plots, you'll plot what's called a scree plot, which tells you for every principal component how much variation is explained. So you can see here that the first, I don't know, 100 or so principal components explain most of the variation. As you get farther out, it gets less and less. You can also look at it cumulatively. Can't talk right now. Cumulatively, yeah. And so I think what this shows you is that it takes a lot of principal components to really get to most of the variation. And I think just keep this idea in mind in the sort of discussion at the end of this. Actually, maybe we won't get to the discussion. We've lost a little bit of time. Okay, so now we got this cool separation, but let's actually see if we can color code this, these different clusters by different population groups. Because see, we have these different clusters, like each one of these dots represents one individual in the data set. And somehow based on their genetics, they were placed into these different areas of the principal component space. And because HD, the Human Genome Diversity Project, provides some population information, you can treat that as a factor, right? These are like the different regions of the world where each of these donors came from.
Cool.
And if you just color them, they actually like pretty cleanly segregate into different population groups.
Nice.
Which I think is super cool.
Yeah.
Like if it's the first time you've seen this, I think it's like, well, that's pretty crazy. It actually works that well.
This is very cool. Yeah.
Yeah, and based on the different, so like on the top left, you have people from Central Africa, and then maybe down here you have like Northern Europeans, Southern Europeans. I guess you also have, right, these people are all have similarly correlated genotypes. Then maybe over here you have Asian, people of Asian descent.
So how strong typically would you expect these groups to be based on, like geography? Or like, is it a strong factor usually or no? Or
yeah, well, that gets into the discussion that I wanted to spend the last part of this talking about. Is I think it's very easy to take this plot and to jump to conclusions and think like, oh my gosh, wow, like genetics really does determine ancestry, and like if you can clearly tease apart different ancestry groups based on their genetics, they must be so different. That's actually not really true at all. If you see here, like the different principal components, they only explain about 4% of the variation that you see in the data.
Right.
So.
And also it looks like in the top left where you have Central Africa, but then you have mixed in, is it Russia? Right? Like a few orange dots for Russian individuals.
I think that's Northern Africa.
Oh, is it Northern Africa? Really?
Yeah, the color's a little bit hard to see.
Okay, the color's a little hard to see for me.
But
yeah,
maybe the other cool thing you can do here, of course, these labels were assigned, like we had the labels, but you could also use another algorithm called k-means clustering, which takes this feature space and tries to find some patterns based on these k-means. Yeah, so in R, the k-means clustering algorithm is built in. It's like just a base function. It works pretty well. So of course, with k-means, you need to decide how many clusters you think exist in the data. So let's say I think there's 3 clusters, and you can, if you're so inclined, you can play around with this later and try different values of k and see what happens. But this can then assign different groups into different clusters. Maybe we think there's 5 clusters here, and it'll give you other clustering groups.
Interesting.
So
have like other groups figured out, and then a larger proportion is like due to, I guess, batch effects or things that might not be in the actual biology.
yeah, I mean, I would hope that batch effects aren't really a factor in this, because with SNP arrays, I think they're quite accurate. Usually you think of batch effects when you're doing RNA-seq and you have like hundreds of millions of reads that little bit differences in how the sequencing libraries were made could potentially influence the results. But the thing with genotypes is that it's very deterministic. Like either you have the variant or you don't. And I wouldn't expect that large batch effects would really be a factor in these differences that you see between populations.
Interesting.
Okay. That's kind of the idea. That's the exciting part. But now for the last 5 minutes, I just want to maybe ruin the excitement a little bit and talk about what this actually means and how we can sort of properly interpret the differences that we see in populations based on genetics. Okay. So I think most people have probably been introduced to this concept through something like 23andMe or ancestry.com, where, of course, these services are empowering to people that might be missing genealogical records, or if they don't know what their ancestry is, they can tell you maybe where their family originated from. But I think they also overemphasize how much genetics actually determines traits or heritage in people's mind. Like, and I'm sure you've seen commercials where it's like, oh, what can your genes tell you about your Spotify playlist? And they'll give you like a playlist based on, you have X ancestry, therefore you must be Y thing. And I think that's kind of not a great way to think about it. Like, of course, ancestry and cultural norms are not determined by DNA. They mean a lot more than that. And while we have definite groupings of different genetic ancestries, it doesn't, by any stretch of the imagination, prove that that race is determined by genetics. Race is, in fact, like a term that we kind of avoid using in genetics. It's more of a term that's been invented. And I think the sort of the biggest reason for this is that populations really aren't as distinct as it may seem on the surface. Many populations are admixed by recent migration events. Like, we know that throughout human history, people have separated, then met up, and of course in history, when different populations meet up, they make babies and just exchange their DNA. Through a plot like this shows you among different European groupings, they each have kind of like different proportions of ancestral populations mixed in. Genetic differences between populations are real, but they tend to be quite small. Of course, what PCA is doing is it's picking up on the small differences that you do find that can separate out, that can explain different groupings. But as we saw from the PCA plot, the first 2 PCs explain about 10% of all the variation. And again, if you go back and play with this, you can plot the 4th, 5th, and 6th PCs, and you can see very quickly the clustering breaks down. Like, there's just not a lot beyond that. And I think And a lot of genotypes really poorly correlate with the things that we think of as defining race. Of course, and a lot of the times people draw charts like this that try to show human evolution as like a tree of different ancestries, but this is really kind of bad or misleading because there is so much admixture between different populations. So the bottom line, what you should take away from this is that race is basically, it's still a social construct. Most traits that we typically associate with race are not really shaped by natural, or are shaped by natural selection. Where skin color is the most obvious example, that it's really shaped by proximity to the equator more so than more evolved people have different skin colors. Like most populations, you can see that from populations in parts of Oceania and that have like darker skin, even though they've evolved from people that maybe had lighter skin before them. It's basically just an adaptation to being exposed to sunlight. And I guess if you're thinking in terms of healthcare, really genetic appearance or racial appearance is not a proxy for disease risk status. And I think that's why why we're really interested in integrating genomics into making healthcare decisions. Because different ancestry backgrounds, like there are certain populations that have higher incidences of certain genetically related diseases. And just basing that that prediction off of someone's like apparent race is really short-sighted in the long run. So the last point is that analyzing the genome is far more useful for making these types of healthcare decisions. And you can see through the sort of like the data science approach, using these clustering algorithms and actually looking at the structure of the data, that this kind of way of thinking really makes the most amount of sense. Okay. Cool, I think that brings us right up to 2 o'clock. So yeah, I hope you guys enjoyed this talk. I know if you have any questions.
It's good. It's really, I thought it was really interesting. Not even just 1,000 random snips still producing.
Yeah.
Mm-hmm. Yeah, but you can also go back and try to play around with the different principal components. And yeah, you can see how there's kind of like the structure dies off pretty quickly.
Right. So I guess, yeah, so the first 2 principal components, I know you were showing that graph, they collectively was about like 10%. So if you took like the 3rd and the 4th, would it be roughly the same, or does it like sort of drop down very, very quickly after that?
Here, well, I can go back to screen sharing to show you what it looks like.
Sure.
Which one was one with the of course. Here, I can make this a little bit easier to play around with instead of just manually like typing them in. What if we plot 4 and 5 and look at the percent variation in principal 1 and 4 and 5?
Cool. Okay, yeah.
Yeah, so now so now like in the Northern Africans, or Central also, just R hacking, move this around so you can actually see. Like, so now this 3rd or this 5th principal component, maybe there's some population structure even within the Central Africans that's a lot less than the first 2 principal components. Like it seems like this 5th principal component is picking up on that.
All right.
Maybe the 4th principal component is picking up on some small difference between, what are these guys? Southern Europeans and yeah, I guess that's kind of interesting. There's maybe the difference that this one population has between the rest of them. I think it's just kind of fun to I guess this third principal component. Then if you go up to like principal component 10 and 11, even though these collectively explain like 1 or 2% of the data, it's like basically nothing.
Yeah, right.
Just a blob.
It's mixed together, yeah.
And these each are like 0.5%.
Right.
Interesting.
Yeah, and I guess maybe the last point I should make is that this PCA analysis is really useful when you're trying to do genome-wide association studies. Because when you're trying to relate a phenotype to, or a genotype to a phenotype, if there is some underlying population structure that could be confounding that association, it's really important that you control for that. And so typically, like the way a genome-wide association study works is you have a bunch of genotypes and a trait, and then a bunch of covariates. It's like a big multivariate regression model. And you'll use the first couple of principal components as covariates in the GWAS setup, which is supposed to account for sort of the underlying population structure.
I see.
Or in QTL mapping, which is something that we do in my lab a lot. Where instead of trying to map like a genotype to a trait, you're mapping genotypes to expression of a gene. You also use principal components as a covariate in that model.
Cool.
Okay.
Very nice.
Interesting. Yeah, very cool.
Okay, cool. So,
all right. Yeah, great talk. Thank you very much. It's really interesting. Good discussion as well. And yeah, thank you again for
thanks for
giving
having me.
Yeah, we'll be happy to have you again in the future.
Yeah, maybe we could do this again.
Yeah, for sure.
Yeah, sorry, with last technical difficulties on my part.
Yeah.
Yeah, no worries.
Yeah.
All right, cool. And then, yeah.